Okay, this is my Unit 3 project, cellular energetics. 3.1 to 3.3 is enzymes. Enzymes are biological catalysts that speed up biochemical reactions. Most enzymes are proteins and they have a tertiary shape that must be maintained to function. This region is called the active site, and the active site has a unique shape and size, with slight changes can occur to align with the substrate. So the substrate is a molecule that can interact with the enzyme, and it has physical and chemical properties that are compatible with the active site, as shown in these images of the substrates binding to the active site. Okay. Next, we have enzymes can indicate the substrate or chemical reaction involved. So enzyme names are ending in ase. Sucrase is an enzyme that digests sucrose. Enzymes are not reusable, and they um, are not chemically changed by the reaction. Cells typically maintain a specific enzyme concentration. Here we have hydrolysis, splitting the substrate, synthesis, binding. Okay, 3.2 enzyme catalysis. Enzymes lower the activation energy requirement of all enzyme mediated reactions, and this is going to accelerate the rate of reaction as shown here without an enzyme, and then with an enzyme, they lower activation energy. All right, environmental impacts 3.3. Enzymes have a unique tertiary structure, as I said. Changes in conformational shape are denaturation. Changes in environmental temperature can lead to denaturation, and pH changes can also lead to denaturation. Enzyme denaturation is typically irreversible, and the catalytic ability of the enzyme is lost or decreased. However, in some cases, denaturation is reversible. A change in the pH outside of the optimum range or the optimum temperature is going to lead to denaturation. And down here we have a graph that shows the optimum temperature will have the highest rate of reaction. And then here are two graphs that show the substrate concentration, that the rate of reaction will increase to that point where all the substrates have bonded, or all the enzymes have found a substrate to bond to. And if is an increasing enzyme concentration, as shown in the second graph, the rate of reaction will continue to increase until one is limited. Okay, enzyme inhibitors. Competitive inhibitors compete with the substrate for the active site of the enzyme. If inhibitor concentration exceeds the substrate concentration, the reactions are going to be slowed. However, if the inhibitor concentration is considerably lower, the concentration reactions can proceed normally. So, regions other than the active site where a molecule is combined are called allosteric sites, and non competitive inhibitors bind to allosteric sites, not active sites. However, this binding is going to cause a shape change, which is going to prevent the enzyme to function because the active site is no longer available and the rate is going to decrease. So, we have two images shown. The first one shows competitive inhibition, and the inhibitor is occupying the active site, therefore the substrate cannot bind to the enzyme. And the second one, the non-competitive inhibitor, is binding to one of those allosteric sites, not the active site, and then the shape is changing and the substrate cannot bind. Okay, now we get to photosynthesis. Big picture, photosynthesis is the process which captures light energy from the sun and produces sugars. Our first light dependent reactions capture light using light absorbing molecules called pigments. The pigments help convert light energy into chemical energy. Chemical energy is temporarily stored as NADPH. So the chlorophylls capture the sunlight energy and convert it into the high energy electrons. And that is the first step, and that is called photoexcitation. As shown here, the chlorophyll is collecting the sunlight energy and is stimulating these electrons. Okay, so the photosystems, photosystem 1 and 2, are embedded in the internal membrane of the chloroplast, specifically the thylakoid membrane. Hydrogen molecules from the splitting of water create a proton gradient, and the electrons from the split reduce the oxidized P680, which we'll talk about. So, stimulating electron travels the stimulated electron from the photoexcitation travels to a reaction center, which is the pair of chlorophyll molecules named P680, and that's gonna boost the electron to the primary electron acceptor, and that's gonna leave P680 oxidized. However, the splitting of water is going to reduce it back to normal, so it can happen again. The boosted electron flows across the photosystems to um, across photosystem 2 electron transport chain to photosystem 1, and this electron flow is going to power proton pumps, which is going to increase the proton gradient. And then due to this gradient, the ATP synthase channel can produce ATP, which we'll talk about. And here's a in-depth or blown up image of photosystem 2. We have the electron going to the P680, the chlorophyll, and then jumping to the electron acceptor and down into the ETC. And then right here, we have a bigger picture showing the thylakoid space and how dense the protons are in the space, and how like the membrane, basically the ATP synthase can operate because of the how many protons are in the thylakoid space. Okay, so photosystem 2 or photosystem 1, electrons flow into photosystem 1 and are boosted to the primary electron acceptor by P700. This is pretty much identical to photosystem 2, which we talked about. And the primary electron acceptor sends electrons to another electron transport chain, and these electrons can then reduce NADP plus to NADPH. Therefore, the light dependent reactions are using light energy in water to make oxygen, which comes from the splitting of that H2O, the ATP from the electron transport chain, and then the NADPH at the end. Okay, here we have a image of photosystem 1, and we have the pretty much identical, except it's gaining one electron right from the sun, which is going to oxidize the P700, and then the electron from the electron transport chain is going to reduce the um, P700, leaving it back to normal, and the P700 is going to excite the electron, and then it will go to the NADP plus reductase, which is going to create NADPH. Okay, electrons flow into photosystem 1 and are boosted. Oh, wait, I already did this slide. Okay, here we have a diagram showing both photosystems. I've talked about everything there. All right. Okay, the Calvin cycle uses products of the light-dependent reactions, the ATP and NADPH we generated, and the CO2 from the atmosphere to produce carbohydrates. And this is going to occur in the stroma. So right here we have a diagram showing the ATP is going to become ADP, donating that phosphate group. And then the NADPH is going to become NADP+, so it's donating a proton, and then that's going to create G3P, which is energy. Okay, 3.6, cellular respiration. Cellular respiration and fermentation are characteristics of all life forms. Cellular respiration and fermentation release chemical energy from organic molecules like glucose. Oxygen is used in cellular respiration and not in fermentation. So, simplified, cellular respiration breaks down glucose and creates ATP. Cellular respiration involves multiple metabolic pathways. Glycolysis occurs in the cytoplasm, and this is going to, every organism is going to do glycolysis because it does not require oxygen. Pyruvate oxidation occurs in the mitochondria. The Krebs cycle occurs in the mitochondria, and the ETC occurs in the mitochondria. Here we have our carbon overview. In the cellular respiration, we're starting with pyruvic acid, a 3-carbon chain. The CO2 is going to release 1 carbon, and then we're going to have 2 carbons as acetyl CoA, and those 2 carbons are going to be released as 2 CO2, and eventually ATP is going to be generated. We'll get into that. Okay. Electron transport chain. The ETC transfers energy from electrons. Membrane proteins make up the ETC. Membrane proteins facilitate a series of coupled reactions using energy from the electrons, specifically from the electron carriers. Higher energy electrons are donated by the electron carriers NADH and FADH, or NAD and FAD. Active transport of protons occurs during ETC reactions, and this active transport of protons establishes an electrochemical gradient across the membrane, which is the most important thing because that's how we're going to generate our ATP, most of it. Okay, oxidative phosphorylation or phosphorylation is the process of making ATP using a proton gradient, and the name basically comes from the NAD and FAD lose electrons, so they're being oxidized, and then a phosphorus is being added to the ATP to create ATP, which is phosphorylation. So that's why the name makes sense. Electrons are delivered by electron carriers NAD and FAD, and ATP synthase uses the proton gradient to create ATP. Decoupling oxidative phosphorylation means ATP synthase will not occur and the protein gradient will generate heat, which can be used to regulate body temperature. Okay, so here's a diagram of the ETC. As you can see, we have our NAD or NADH becoming NAD plus, donating that electron, which is helping to induce the gradient. Same thing happening with FADH2 is donating an electron, and those are going to travel across those proteins embedded in the membrane. And ultimately, this is just going to increase the gradient, which is going to make ATP synthase possible, which will produce ATP. Here is our net ATP. We invest 2 in glycolysis and get 4, so 2 net, and then 2 from the Krebs cycle, and then 32 from that electron transport chain. So a total of 36 ATP, around 36 ATP generated. Okay, here we have fermentation, or anaerobic respiration, or so the oxygen is the final acceptor in the ETC. So if oxygen is not available, you can't go through that process. So it's going to be anaerobic respiration, and it's either going to be lactic acid fermentation or alcoholic fermentation. The alcoholic fermentation is going to yield 2 ethanols and 2 ATP, and then the lactic acid is going to yield 2 lactate and to ATP. All right, that's all I have. Thank you.